Hello everybody, thank you for coming to my poster presentation. My presentation will be on the application of mixture models to RNA-seq data to understand aging regulators. My name is Sas and I'm from the University of Queensland. So before we go on, I want to give a bit of background on aging and why we're having so much of problem trying to understand what causes aging. So the main reason here is because aging is heterogeneous and it's also multifactorial. And we've only recently discovered that only about 20% of our genes dictate how much or how well we age. Everything else is a whole host of other reasons. And furthermore, to make matters even more complicated, we don't age. As an organism, we don't age constantly. So that means different parts of our organs age at a different levels and different rates, and therefore it makes it a bit more harder to understand what is the main cause of aging. And so therefore a lot of people have come up with multiple different um, ways to measure and to understand aging, and these are called all hallmarks of aging. As you can see, there are a couple listed over here. But before we go on, I also would like to state what the goal is of studying aging. So the goal of studying aging is not to live longer, but to find ways to intervene so that we can live healthier longer. So this is where mixture models come in. Mixture models are extremely good at describing heterogeneity within a population. So for example, figure A is quite straightforward. We can see the differences between the mean while the standard deviation is the same. The figure B here, we can see that the mean is still the same, but the standard deviation is slightly different. And once again, we have methods to develop that and tell that differently as well. There's also one more quite obvious as well. Last but not least, we have panel C here, where the mean and the median are quite similar to one another, and that makes it a little bit harder to tell variation and differences between these two populations itself. And therein where mixture models come in. So mixture models are really good at differentiating these two specific criteria because they can incorporate multiple different models and distribution, Gaussian and non-Gaussian, and they're really good at discovering unobserved subpopulations without requiring the data to be labeled. And furthermore, once we have our data assigned to particular models, we can then make sure the models fit are the best particular models, and therefore these particular models can then be determined by the Bayesian information criteria. So first thing we did was we simulated a whole bunch of gamma distributions, and we then used three separate packages to determine M class, R mixed mod combi, and mixed all gamma. And so all you can see, a majority of all the genes that were detected in terms of being the best model assigned. Majority of that was R mixed mod combi, and a couple of them were determined by M class and also with mixed all gamma. So now that we have done that, we've decided next to move on to determining and to test whether our hypothesis is correct, meaning that we can then cluster genes in terms of their subpopulation. And for this, we move to the PAM50 genes. So really briefly, PAM50 genes are genes, the 50 genes that have been determined to classify specific types of breast cancer. So here we have three separate types of breast cancer. As you can see, a majority of them has been clustered based on the specific types of cancer subtypes that they have. Except for these three, we can obviously see that these three were meant to be Loom B, and you could see a majority of clustering of Loom B here. However, there are also major clustering for both, for specifically for basal subtype as well. That's one of the main issues that we have, meaning that this doesn't mean that this doesn't work. It means that there may be specific basal criteria within these specific types as well. So this is the pipeline that we've developed. We have RNA-seq data. Specifically here, we'll be using the GTEx version 7. We'll normalize it using the YARN methodology, and then we'll run these three specific mixture models, and then we'll end up coming with the best model. Once we've done that, we'll run an exact test to determine whether the genes are significant, and next we'll also run an exact test specific for gender as well. And then if any of these genes are overlap, we would like to be a little bit more cautious and state that these genes were then mainly based on gender, and then you will remove them. So once we have them, we'll then go on to a downstream processing where we will measure both between aging and traditional methods and also non-traditional methods. And traditional methods here would be the HR genes. So one of the main challenges specifically with mixture models in terms is that we wanted to make sure that we can determine a total number of modes. And so as you can see, we ran a large number of them here on most of the data, and then we were able to determine what are the number of modes that are most commonly occurring, and these were 1 to 3, and therefore we use them moving forward. So not to be surprised, we also found out that the majority of the genes that were determined and detected was specifically from RMix Combi, matching for what we noticed previously with our own simulated data as well. So one of the things that the key takeaway here from everything else from this entire lecture is specifically that mixture models have detected unique genes and also within similar known pathways. What here I wanted to show is the total number of mixture models unique genes detected in comparison to HR. As you can see, some of the tissues did not have any specific genes that were detected. And most importantly here, we have least number of common pathways, which is exactly what we were after, because the goal or the idea here is that we'll be able to discover genes that were not previously detected. Once we have that, we then compare the genes to known data sets. So we have the Digital Aging Atlas and GenAge. So these are the total number of mixture model genes and total number of HR genes. And the genes that overlapped with all the particular databases was 56 and 66 for HR respectively. And also mTOR was the only gene that was unique between mixture model genes and HR genes. Unfortunately, one of the things that we've noticed as well in particular is that mixture model genes in particular seem to be correlated with the sample size, meaning that we get more genes on the larger the sample size is, and therefore this could be one of the limitations that we have in mixture model genes in particular. And so lastly, to sum up, it's important for us not to assume a single distribution or assign a single mode for transcriptomic data specifically within the gene levels itself. Ideally, this mixture model should be used in conjunction with different gene expression methods such as HR. Furthermore, mixture models for aging RNA-seq data seem to have three very distinct advantages. Firstly, as I said, it's ideal for heterogeneous data. Secondly, it allows us to test out different distributions and select the one that fits best. And last but not least, clustering and cluster numbers are determined in almost an unsupervised manner. And last but not least, one of the disadvantages for mixture models is its high computational cost, and therefore it also might require larger data sets.